Hi dear students, a warm good morning to all of you. Welcome to the next topic, bioremediation. In the previous sessions, we had mentioned about various synabiotics, uh, what are the effects on the environment, what are the effects on human body, and it's quite clear that we need to find a way how to degrade them, okay, or make them less harmful. And for that, we use different methods. And one among that is called bioremediation. Now, let me just explain to you what do you mean by the term degrade, remediate, and all that. The term remediate, degrade, and all that means if you have a 7 carbon compound, you have to break it down and make it into smaller carbons so that it becomes a single carbon molecules, for example carbon dioxide. Now there are substances which cannot be remediated or which cannot be degraded. They are of course persistent organic pollutants which we have, but there are certain ones which could be degraded or remediated also. So you understood what is the meaning of remediation or what is the meaning of degradation also. But now let's link that with the word bio. Bio means any living agent. So when you use any living agent to degrade or remediate a organic pollutant, then that is called as a bioremediation. Bioremediation is thus the application of any living organism. It can be any large organism, okay, living organism, or it could be any what small microscopic organism also. Now, for example, I can tell you something. If you might even use earthworms sometimes to remove certain organic pollutants, or sometimes you might even use bacteria to remove certain xenobiotics, or you can also use plants for the remediation of organic substances. So when you use any living organism, let it be plant, or it could be any other living form, or it could be a macroscopic form, or it could be a microscopic form, then it is referred to as the process of bioremediation. And even you can see that now, even in the case of plastics, we try to remediate them using various insects so that if they could be broken down or being degraded into smaller particles. And you can see that various enzymes from such in the insects there were certain microorganisms, and it is a microorganism which produce enzyme to degrade plastics. Now you have a plastic degrading organism called like Ideonella sacaiensis. It is a PET degrading. That, you know what is PET? PET bottle and all that. It's a plastic bottle. The PET degrading enzyme called the PETase is mainly being produced by Ideonella sacaiensis, which is a microorganism. So, when you use living organisms to degrade or detoxify the pollutants, you call it as bioremediation. Now, why do we have to use this? Usually, such bioremediation technologies are being considered to be as efficient, economic, versatile, and also environmentally sound technique. And I would be more predominantly concentrating on the microorganisms for bioremediation, mainly because this is a microbiology lecture which I am giving out. And so microorganisms usually they can mitigate and degrade or even reduce the hazardous effect of organic pollutants so that they will be converted into innocuous compounds such as carbon dioxide, methane, water, and biomass without adversely affecting the environment. So do remember, the term bioremediation doesn't mean that it is only the microorganisms. It could be others also. But since I am dealing with microbiology, I am concentrating with the microorganisms which are capable of degrading the hazardous organic pollutants. Now, In 1972, there was the first commercial use of a bioremediation system, mainly to use a cleanup of a sun oil pipeline spill in Ambler in Pennsylvania. So let me just talk about that. When oil spills happen, okay, usually what is the process? Either you can burn out the oil which is there, but of course that causes again pollution, because you are causing harming the ecosystem within the water. Of course, oil has been obtained now in oil ridges and all that, so there'll be oil which is spreading all over that particular place. Or they might put like vents, they might not vents, they might put rope like structures so that the oil will not spread to different areas. These are all been things which are being carried out, but it's not easy to remove the oil from there. So the concept of using microorganisms, especially oil eating microbes, were being utilized over there. And one famous thing when we talk about the oil-eating microbes is the discovery of Chakrabarti's bacteria, which is Pseudomonas, a strain of Pseudomonas putida. Pseudomonas is a green color bacteria, and okay, there are different Pseudomonas. So Pseudomonas putida, okay, Pseudomonas putida is Chakrabarti's bacteria, which was artificially produced to contain large amount of plasmids in it, so that it was considered, it was producing a, no, it was capable of eating the oil at a very faster rate. Okay, and that organism was patented also, and so that's about the first use. Like, we have oil eating bacteria in the process of bioremediation. Now, if you go to consider bioremediation, especially with the microbes, it can be carried out in two different ways. One is you can apply the microorganism, okay, at the site of the contamination itself. Now, for example, as in the case of oil spill, in the sea, in the ridge, when a oil spill is happening, it is not possible for you to take the whole water from there and then go and remediate it using a microorganism. So what you have to do, you will have to put the microorganism at the site of contamination, so that the microbe will eat all over the organic pollutants. That is the what that is the in situ remediation. And coming to the next one is the ex situ remediation. Now, what is ex situ remediation? Ex situ remediation is nothing but you will take the contaminant from the site of the contamination and treat it elsewhere. Now, for example, if I have I'll tell you, if I have some sand which is being contaminated with heavy metals and all that, I can take that sand, dig out that sand, and take it to another place and remove the heavy metals from it. That is a ex situ remediation. So, bioremediation can be done in situ or ex situ, and there are different methods by which you can do it, like bioaugmentation, composting, bioventing, biopiling, biosparging, natural attenuation. These are all just methods, technical methods. They differ in only their technical means, but they are different methods to do the process of bioremediation. Now, in this process, either microorganisms will be added to them so that the bioremediation will happen at a faster rate, or we will add some favorable environments for the already present organisms in the soil or in the environment, so that they will promote bioremediation. So bioremediation can be in situ or ex situ, and in situ remediation, you apply it at the field. Either you can use the microorganisms which are already been present over there, or you can give a what engineered bioremediation, in which different methods are being given so that the organisms, what is it, will be promoted to grow over there at a very faster rate. Okay, and different biosparging, bioventing, these are all methods which will help to what increase the growth of the organisms and it is an engineered, because we are using engineering aspects, we call it as an engineered bioremediation. Now, the other one is the ex situ remediation, where what the organic pollutant or the pollutant or the what pollutant do you have? You take it from that particular site, take it from over there, and then you do the remediation at a different place. Either you will do it in a reactor, or you might do it in a solid, that is without making it slurry, means make it into a watery type form and then you remediate it in a bioreactor, or as such itself, if you do not treat it and if it is in the solid stage then you remediate it, it is called the solid phase bioremediation, and you have different methods over here. So now we are going to explore what are these in detail. So in situ remediation, you should have, I told you it is on-site remediation. So either you should have sufficient microorganisms which are already capable of remediating to bioremediate the contaminants, and you should have nutrients so that the microorganisms will grow. You should have a good environmental condition, and sufficient time should be allowed so that the natural process or to degrade should be present over there. Now, if you do not have sufficient microorganisms to be there, you can also add it over there, so that, as I told you in the case of oil spill, they added Pseudomonas butida so that the oil spill was being degraded. So you need all these things for the in situ remediation. So let's go into the details of the various techniques which we have. One thing what we have to say is about, before that I'll just go to the now in situ remediation. In situ remediation can be done by bioventing, in situ biodegradation, bioparging, bioaugmentation and all that. These are different methods. So we'll start with bioaugmentation. Bioaugmentation is nothing but when indigenous or exogenous microorganisms are frequently added to the contaminated site in order to reinforce the natural biological process. And in this method, specific competence traits or a consortium of microorganisms are introduced to improve the degradative capacity of a contaminated area. So, let me just tell you, if we have a contaminated site, okay, contaminated site, now for example oil spill, sometimes there would be oil degrading organisms already being present over there, okay, or in another case you can add a competent strain which is capable of degrading oil, or you can also add a consortia of microorganisms. What do you mean by a consortia of microorganisms? A group of microorganisms which can be grown together and which can be having which are capable of doing the particular function. Now, for example, if I take A, B, C, D, E, 5 organisms which are capable of degrading oil, and I grow them together and put them together at an oil spill, that is referred to as a consortium of microorganisms which is being used to degrade the contaminated area. So either you will add some nutrients or something so that the organisms which have been present at that particular site will be promoted to grow so that they can degrade the pollutants, or you will add a consortium into the site so that they will be promoted to grow then. And bioaugmentation is more commonly and successfully used at contaminants used on contaminants removed from the original site, such as the municipal wastewater treatment facilities, and they can be used in such cases. And it is used at sites where soil and groundwater are contaminated with chlorinated ethenes such as tetrachloroethylene, trichloroethylene to ensure. At two different ways also you can use it, either in situ as well as ex situ also you can use. Either in wastewater treatment plants, you take the water and put it into the wastewater and there you use it, or if where the soil or the groundwater is contaminated, you will have to use in situ microorganisms. In such cases also you can use the process of bioaugmentation. So in the process of bioaugmentation, you are promoting the growth of either the indigenous means the organisms which are already been present, there in that environment. And exogenous is all those microorganisms which are added to the environment to promote the organic pollutant degradation. Now the next one is bioventing. This is an in situ treatment method. Okay, and here what you do is you add air and nutrient supply through wells to the contaminated soil so that the indigenous bacteria will be uh, promoted to grow. You just remember in ACs we do have vents, okay, and the same way here in the soil you will prepare some vents, and through the vent you will produce air as well as nutrients into the soil so that the organisms which are capable of degrading the pollutant will grow over there, and they will, what they will do, they will degrade and reduce the toxicity of the pollutant and prevent its volatilization or reduce its volatilization. Volatilization is nothing but its ability to become vapor and escape. So to prevent their escaping to the atmosphere is being carried out. You can do the process of bioventing over there, and it works for simple hydrocarbons and can be used where the contamination is under the deep surface. For example, you can look into this. Using a vacuum pump, you will put what, air, and as well as you can also add some nutrients also onto this thing. So when you add air and nutrients into this, the organisms which are already been present in this environment will degrade the what, the will degrade the organic pollutants present in this contaminated site. This is a case of bioventing. This is another explanation of the same thing which I just included the figure for your reference. So hope so that is clear, and let's move to the next one called the biosparging. Biosparging for increasing now groundwater oxygen concentration and hastening the rate of biological degradation of contaminants by indigenous microorganisms. The injection of air under pressure below the water table is done in this method. And what happens is it increases the contact between the soil and the groundwater through mixing in the saturated zone. And biosparging is commonly used at sites with mid-weight petroleum products such as the diesel fuel. Now, for example, now let me just explain it a little bit more. Okay. Sometimes when it goes to below the water table, okay, below the water table, what happens? The oxygen level will be found to be very much less. Okay, and so the biological degradation of organic pollutants will be at a very low rate. But you know, if an organic pollutant happens into enter into the groundwater table, that is going to affect the entire water tables of the water bodies of that particular area. So in such cases, what you do is you will provide oxygen at a very low level, okay, so that the organisms which are present over there will grow and try to degrade what, the pollutant over there. Now just have a look at this. This is a case of biosparging. That is, the difference which we have here is the using a compressor or a vacuum pump, you're going and giving the oxygen below the what, it's a little more deeper. In the previous bioventing case, what does it means? You're just giving it at the surface level or to some level, but you do not go to expect it to come to the groundwater level. So in such cases, it goes a little more deeper in the case of biosparging. And what happens when you give oxygen and everything? Those pollutants which could happen to enter into the water table also could be prevented. Like when the organic pollutants happen to enter the groundwater table, there itself they have to be degraded. Okay, so to prevent that, we do the process of biosparging, where a high vacuum pump has been used to provide large amount of oxygen. And biopiling, this is in this method, the contaminated soil is excavated and mixed with the soil amendments and then placed on a treatment area where the bioremediation is done using forced aeration, and carbon dioxide and water are produced after the contaminants are reduced, and a treatment bed, an aeration system, an irrigation or a nutrient system, and a leachate collection system is required for the biopiling system. And of course, to enhance the biodegradation process, moisture, heat, nutrients, oxygen, and pH all are being controlled. Now, just let me just help you. You take what is a biopiling system. You don't have to get confused looking at this particular picture. It's nothing but you dig out the soil from the contaminated soil, you see, and then you will put it at a particular place where what uh, the pH, environment, and all that is being made favorable. That is, the microorganism is promoted to grow. All the nutrients, the environmental conditions are all made being favorable, and hence the what it is being piled at a particular area, and it is being degraded. So, using certain engineering techniques, we are trying to make sure that the organism which is capable of degrading the soil can grow at a very faster rate. Then the other one is composting. Composting is a term which is quite familiar to all of you. Here, the contaminated soil is combined with a non-hazardous organics amendments. Now, for example, manure, agricultural waste, and all that, and then it is being composted. And as a result, the rich microbial population is developed in the presence of these organic materials and elevated temperature. And during composting, what happens? They will transform the degradable organic waste into humus-like substances. So it's nothing but when you add manure like cow dung and all that, as we do in the biogas, in composting, when you add some manure or agricultural waste along with the organic pollutant containing soil, what happens? The organisms which have been present in the manure or which are present in the soil, utilizing the nutrients, they will grow rapidly, and the organic waste is being converted into humus-like substances. These are some cases at which what does it mean? You'll put different types of thing, and you will put organic waste also on top of them and allow them to degrade, and you will just it's closed. This is explosives are being removed during the Windrow composting. That is a picture. Large amount of explosives were there, so they were being mixed with uh, soil as well as other organic wastes, and the composting was being done. A picture of that. The source is also being provided. So composting is a process by which the organic wastes are degraded by microorganisms, typically at elevated temperatures. And the typical compost temperatures are in the range of about 55 to 65 degrees centigrade. And at such increased temperatures, what happens? The heat produced by the microorganisms the elevated temperature is mainly because of the heat which is being produced by the microorganisms when they are being trying to degrade the organic materials. Now, this was a windrow composting. You might have noticed this is a windrow composting which I had mentioned, and in windrow composting, it's just in higher extension. You just have to know it. I'm adding it for your knowledge. The contaminated soils are excavated and screened to remove the large rocks and debris, and they are being transported to a composting pad with a temporary structure to remove containment and the protection from the weather extremes. And then of course you will undergo some changes. Now, for example, amendments are called changes. You can add straw. I told you manure or agricultural waste can be added. It can be straw, alfalfa, manure, agricultural wastes, wood chips, etc. They are all mixed together. And the soil and the amendments are layered in long piles called the windrows. And the windrow is thoroughly mixed by turning it with a commercially available windrow turning machine. It's just like tilling your field, just like that. These things are all being together mixed using a windrow turning machine. And the moisture, pH, temperature, and the explosive concentrations, they are all being maintained. Of course, if the organic pollutant is at a very high level, the microorganisms will be killed. So you have to maintain the explosive concentration at a level which is permitted, and leave them for some time so that they will get degraded. And that's, if you want to see the image, you can just go through this site. I have just put a link. And the other one is land farming, where you excavate the contaminated soil, and after that the soil is spread over a prepared bed, and which is already previously tilled, and you will stimulate the indigenous biodegradative organisms to grow over there, and you will also plant some plants also, and land farming is being carried out so that the plant will take up all the organic pollutant rather than from the soil. For example, in a study, of course I can't tell you the source, but if you go to look into the Kerala environment, you can see that lots of chira. Okay, chira is a green leafy ones which we have. Okay, the chira is being found to be a very good what do you say, pollutant absorber. It will take large amount of heavy metals which are being present in the soil if you grow chira in that particular environment. So the into the plant, this is a case of phytoremediation also. Okay, where the plant is also being taking the pollutant from the soil, and the organism of course is also trying to degrade the different types of pollutants which are being present in the soil. So what are the salient features of bioremediation? It is a biotreatment process for contaminated sites such as water and soil, and microorganisms can degrade the contaminant and increase their number in the presence of a contaminant. So the microbe is increasing in number and trying to reduce the organic pollutant. And this organic pollutant can either be degraded into what? Harmless products like carbon dioxide, methane. Of course, methane is caused global warming, but they will be reduced to smaller forms which are being found to be less toxic or less harmful or sometimes even harmless components. And the degradation is being augmented with the help of the microorganism. And of course, when the microorganism population is being declined or reduced, the degradation of the contaminant also will be found to reduce. And this can be done in situ with less efforts and often without causing a major disruption of the normal activities. Just as I told you, in oil spill, you just have to put the bacteria over there. The bacteria will run over the sea and eat the oil spill, and it also helps in the destruction of the complex pollutants, and many of the hazardous compounds can be transformed into harmless simple products. Thus bioremediation can eliminate the future liability of treatment and disposal of contaminated material. Now you might think I have added a lots of engineering aspects into the bioremediation into it. These are all methods to what increase the rate of biodegradation. Normally, if you just rely on the microorganisms alone, the time factor is a factor. So by incorporating the engineering aspects also, we will be able to increase or fasten the degradation, and less energy and manual supervision is required, and does not require the use of any hazardous or toxic chemical. And of course, it is limited because it is limited only to those compounds that are biodegradable, and it is highly specific. And there are some concerns that the products of biodegradation may be persistent or toxic. Of course, that we need to screen, and we need to check in the lab whether the pollutant after degradation, whether it will be more toxic or to say only those microorganisms we can use. And you should also give suitable environmental growth conditions for that organism to be grown. And it is difficult sometimes to extrapolate from bench and pilot scale studies to a full scale field operations. That is, from the lab to the field, sometimes it might not give you the same results itself, to a lower level might be, but you can extend bioremediation to successfully in a successful level by proper research and as well as clean site techniques which we need to follow. With this, we come to an end. Thank you.